Welcome to this lecture on primate phylogeny. Today, we are going to embark on a fascinating journey back in time to trace the human family tree. We will explore exactly how we are connected to monkeys and apes and discover the incredible scientific tools used to map our evolutionary history. Understanding this topic is not just about knowing the past, it is about understanding who we are as a species today. To begin, imagine you are attending a massive family reunion where thousands of relatives have gathered. You would naturally notice that you look very similar to your siblings, somewhat similar to your first cousins, and only vaguely similar to distant relatives. This exact same concept applies to the animal kingdom, specifically to primates. Primate phylogeny is essentially the ultimate family tree for our specific branch of life. It maps out exactly who our closest evolutionary cousins are, like chimpanzees and gorillas, and traces our lineage back to a single common great-grandparent that lived millions of years ago. By looking at this tree, we can see the step-by-step journey of how a small, tree-dwelling creature eventually gave rise to modern humans, walking on two legs. Now that we have an intuitive grasp of the family tree, let us look at the formal scientific definition. In evolutionary biology, primate phylogeny refers to the rigorous study of the evolutionary history, development, and relationships among various species within the order primates. This order is broadly divided into two main suborders. First, we have the Strepsirini, which includes lemurs and lorises. Second, we have the Haplorini, which includes tarsiers, monkeys, apes, and us humans. To construct this complex phylogenetic tree, scientists do not just guess. They use a powerful combination of physical fossil records, comparative anatomy, and modern molecular biology techniques like DNA sequencing to piece together exactly how and when these different groups branched apart. Building on how these trees are constructed, let us zoom in on our own specific branch. According to this evolutionary map, the lineage leading to modern humans diverged from our closest living relatives, the chimpanzees, roughly 6 to 7 million years ago. The sequence progresses from early ape-like ancestors, such as Dryopithecus, through various hominin stages like Australopithecus and Homo erectus, ultimately culminating in anatomically modern Homo sapiens. Understanding this sequence requires analyzing how specific traits emerged sequentially over millions of years. Two of the most critical traits scientists track are bipedalism, which is the ability to walk upright on two legs, and a significantly increased brain size that allowed for complex tool use and language. Knowing our evolutionary history is fascinating, but you might wonder why scientists spend so much time and resources figuring this out. The answer lies in both understanding our past and protecting our future. Because chimpanzees and macaques share a vast majority of their DNA with humans, they respond to diseases and medications in ways that are incredibly similar to us. This phylogenetic closeness is exactly why certain life-saving vaccines and treatments are developed by studying these specific primates, rather than unrelated animals like mice. Furthermore, understanding primate phylogeny is absolutely crucial for wildlife conservation efforts. It helps biologists identify genetically unique, endangered primate lineages that need immediate protection to preserve the Earth's evolutionary heritage. To figure out exactly when these branching events occurred, scientists use a brilliant mathematical concept called the molecular clock. Conceptually, this relies on the fact that DNA mutates at a relatively constant rate over millions of years, acting like a steady ticking clock inside living cells. The formula we use is T equals D divided by 2R. Let us break down these symbols. The letter T represents the time that has passed since two species diverged, usually measured in millions of years. The letter D stands for the genetic distance, which is the total number of DNA mutations that differ between the two species today. Finally, R represents the constant mutation rate per million years in a single lineage. We multiply R by 2 in the denominator because both lineages have been mutating independently since they split. If the genetic distance D increases, it means the species diverged a much longer time ago. Now that we have our formula, let us see this with a concrete example by calculating the human and chimpanzee divergence time. We're given that scientists found a genetic distance of 0.012 between the two species for a specific gene. We are also given that the mutation rate for this gene is 0.001 mutations per million years per lineage. We need to find the time T. We start by substituting our given values into the molecular clock equation. In the denominator, we multiply the mutation rate by 2, giving us 0.002. This accounts for the mutations accumulating in both the human and chimpanzee lineages independently. Finally, dividing 0.012 by 0.002 gives us exactly 6. This means humans and chimpanzees diverged approximately 6 million years ago, a result that perfectly aligns with the fossil record. So we have successfully used the formula to find time. This brings us to an important question. How do scientists actually determine that mutation rate in the first place? We can figure this out by working backward using fossil data. In this first chunk of our derivation, we are setting out to rearrange our molecular clock equation to solve for the unknown mutation rate, R. We start with our original equation, T equals D divided by 2R. To isolate R, we first multiply both sides by R, moving it out of the denominator. Then we divide both sides by T. This algebraic rearrangement shows that the mutation rate is equal to the total genetic distance divided by twice the divergence time. This new form of the equation is incredibly useful whenever we have a reliable fossil to anchor our timeline. Now that we have rearranged the formula, let us apply it to real fossil data to continue our derivation. Paleontologists have discovered a well-dated fossil showing that Old World monkeys, like macaques, and apes, like gibbons, diverged exactly 25 million years ago. This gives us our time, T. Geneticists analyzed their DNA and found a genetic distance of 0.050, which gives us D. We plug these known values into our newly rearranged equation. The genetic distance, 0.050, goes in the numerator. The divergence time of 25 million years goes in the denominator, multiplied by 2. The mathematical reasoning here is that we are taking the total genetic difference and spreading it across the total time both lineages have been evolving independently. Let us complete the final steps of this derivation to find our calibrated mutation rate. We simplify the denominator by multiplying 2 by 25, which gives us 50. This number represents the total millions of years of independent evolution that have occurred across both branches combined, 25 million years for the monkey branch, plus 25 million years for the ape branch. Dividing 0.050 by 50 gives us a final mutation rate of 0.001 mutations per million years per lineage. The intuition behind why this works is that we have successfully calibrated our molecular clock using real-world fossil data. we can now confidently use this specific rate to date other primate divergences, where no fossils are available to guide us. Having proven how the molecular clock works, let us look at the complete picture it helps create. What you are seeing here is a comprehensive primate phylogenetic tree. The horizontal axis at the bottom represents time, starting from 65 million years ago on the left all the way to the present day on the right. As we move from left to right, you can see the main trunk splitting into various branches. The earliest branches represent the prosimians and tarsiers. Further along, the monkeys branch off. Finally, we reach the ape lineage, which sequentially branches into gibbons, orangutans, gorillas, chimpanzees, and humans. Notice the specific node where chimpanzees and humans split? It aligns perfectly with the 6 million year mark we calculated earlier. This diagram beautifully summarizes millions of years of evolutionary history into one clear visual map. We have covered a lot of ground today, so let us review the most important ideas from this topic. First, primate phylogeny is the ultimate biological map that traces our evolutionary journey from a shared ancient ancestor. Second, we learned how scientists reconstruct this history by combining the physical evidence of the fossil record with the mathematical precision of the molecular clock. Third, this knowledge is not just historical trivia. It provides critical insights for modern biomedical research and wildlife conservation. Finally, understanding this family tree reminds us that humans are not separate from nature, but are deeply and genetically connected to the rest of the primate world. You now have a solid understanding of how we trace the human family tree.